So this is the second of the lecture for prep videos, and we'll talk about the initiation process at the level that you need to understand it. There's going to be more details in the textbook and some of the resources that are given for you to use as preparation. RNA polymerase, let me just turn on the pen here. I'll show you what you need to know about RNA polymerase in a minute. But it basically binds to special DNA segments, and these are referred to as promoters, and they are positioned near the transcription start site of each gene. So you have a gene, which is a piece of DNA, and there will be a couple of important sequences that are required for the RNA polymerase to bind to start the transcription process. And this arrow is one of the shortcuts for expressing that a gene is being transcribed. So the RNA polymerase is going to move this way along the DNA, and it's going to make an RNA from 5' to 3'. And in bacteria, if you look at the sections upstream, that means just before, upstream of the coding part of the genes, you'll find that there are two locations where almost the same sequence is found in all genes. They're not identical, but they are highly similar to each other. And then once the RNA polymerase has bound stably at these positions, then transcription is begun. So it's about finding and binding to the correct promoter sequences. And I'm not sure if we have time, but we may talk later a little bit more about eukaryotic gene regulation. And if we have time, you'll find that there's a lot more complexity in how eukaryotic genes are turned on. They have the same kind of core promoters. They're not spaced the same way. And they have many genes have additional regulatory sequences and different kinds of proteins activating and repressing gene expression. We may get a chance to talk about that later. For now, this is what you need to get a good feel for before we proceed. So if we're looking just in general at sort of the gene, a gene consists of the coding information, but also all the regulatory sequences, or the sequences that allow for the activation of the gene's expression or the turning off of the gene's expression. So this is some sequence, and this is a few different genes, 7 different ones here from E. coli, which is a type of bacteria. And then the different stars that are shown up here on the schematic shows you what they're referring to on the actual sequence. So you get a better sense of what we're really talking about. We draw these diagrams a lot just using sticks and lines and arrows and so on. But what we're intending you to understand is that, this line is referring to DNA sequence. And if you were to look at the DNA sequence ahead of, so this purple star is where transcription starts. So the first nucleotide that gets made is going to be different for all these different genes, right? But if you were to look 10 nucleotides back from the start of transcription, you will find that the sequences are very similar, and they're called Tata boxes because what is called the consensus is TAT-AAT. And when we say consensus, what we mean is if you look at the first position here, most of the time you're going to see a T. If you look at the second position, most of the time you're going to see an A. If you look at this position, most of the time you're going to see a T, but sometimes you will see another nucleotide, and so on. So if you go through here, you'll see that there's a little bit of variation. So occasionally there's one that has 1 or 2 different nucleotides, but for the most part, the sequences are the same. And this is almost identical, except the T and the A are reversed. And so that's the Tata box. And then there's another region at minus 35. So that means 35 nucleotides up before the beginning of the transcription site. And you'll see again, it's not quite as strongly conserved. Conserved means they have the same sequence, but this is the consensus. And so the ones that are highlighted are the ones that are most commonly found. These randomly chosen genes are useful for us to look at because you can see that there's places where the sequences are not quite identical, where there is some variation. But notice that especially at the very beginning of that sequence, almost all of them have identical sequence. And the same with the Tata box. These two nucleotides at the beginning and these two at the end are the most strongly conserved. So these ones are seen most often, and then there's more variability in the middle. So all of this is just to say that those are the two sequences that are used by the RNA polymerase to bind correctly to. Notice how in the other parts the sequences are hugely variable. There's a lot of different nucleotides in and around here because they're all different genes. So when we see sequences that are very similar to each other in a lot of different genes that are encoding different proteins, then this tells us that these are functionally important. And we know, for instance, that if you were to delete the promoter, because this is what we call the core promoter, if you were to delete these sequences from the gene, there would be no transcription of it, because this is what the RNA polymerase binds to in order to start the transcription. So just these are some prompts for you to think about, and then to look and just kind of see what you can see about these sequences. I will erase all my stuff here. Yeah, and so this will help you think about and feel more confident about your understanding of what these sequences are for. Okay, and so this is the kind of thing that you may see. A basic gene includes the coding sequence, the transcript. The transcript is longer than the coding sequence for eukaryotes and prokaryotes, but much more so for eukaryotes. The gene is basically the sequence that's going to be transcribed plus all regulatory sequences, which includes sequences to either side of the transcription unit. And then when the transcript is made, there is usually sequence at the beginning, a little bit at the end that is not translated. You have to have binding sites for ribosomes and so on for the transcript. And then the part that is actually the coding sequence that gets translated to make the protein is even smaller than that. And just to note that in eukaryotes, there are little spacers inside the coding sequence so that some of those have to be cut out. They don't contain amino acid information. And so you can sort of see the layout of a basic gene structure, and just notice that the transcription start and stop sequences or signals are different from the translation start and stop. Transcriptional start is basically wherever the RNA polymerase finds those core promoter sequences. It's a certain distance away from that, 10 nucleotides from the Tata box, and that's where transcription begins. Translation start and stop is based on the nucleotide sequence, and you'll see more about that when we talk about translation in the next couple of lectures. And so just one of the things that you can get from this picture, this is just a summary, is that the transcript has regions that are not translated, and this is more the case in eukaryotes than prokaryotes. And when you're thinking about, so this is just the transcription unit, TU is what we call it. So that is what gets transcribed, and there are regions on the 5' end of the RNA and on the 3' end that are called UTRs, untranslated regions, and these are required for things like stability, sometimes for localization of the sequences, sometimes for other regulatory purposes. At the 5' end, there's going to be signals for the binding of ribosomes and so on. And there's also introns, which we'll talk about later. So of this larger sequence, only a subset of the nucleotides in there is going to provide amino acid information to make a protein. Okay, so we're only at this point going to talk about prokaryote transcription, just the mechanics of it. As I said, we may have time to talk a little bit about eukaryotic gene regulation later, although if you take the appropriate MBB courses, you'll get so much detail about this that you'll have sufficient information. So the RNA polymerase, there's a name, it's called holoenzyme. And what this means is the holoenzyme is the enzyme that is constructed of a bunch of different subunits. So there's 2 beta subunits and 2 alpha subunits. And they form a tetramer, two dimers together to make 4. There's a little factor called the omega factor here. And then there's the sigma factor. And the sigma factor is important because it's the one that actually recognizes and binds to that Tata box in the other sequence at -35. And because of the way it binds to the DNA sequence and then to the rest of the protein, or proteins, I guess you'd say, it's responsible for the correct positioning. And it also leads to the separation of the DNA strands, which allows for the transcription to start. So everything's sitting there, the sigma factor binds to the correct sequences, the DNA opens, and when transcription begins, the sigma factor gets released. And then it can bind to another set of subunits and start initiating transcription somewhere else as well. Okay. So this is the rest of the initiation process that we were talking about. Now the elongation stage is just the production of the RNA, and it's called iterative because it is a repeated set of steps. A nucleotide is added, and then another nucleotide, and then another nucleotide. The DNA is unwinding ahead of the enzyme and closing again behind it, and then the RNA that has been made is kind of pushed aside, displaced to make a single-stranded RNA emerging from the transcription bubble. Okay, so this is the elongation stage. And then there's a number of, there's 2 main mechanisms. We're going to talk about the intrinsic mechanism. This requires terminator sequences that are actually part of the DNA and then get transcribed. And so you have a stretch of 40 base pairs that has at the end of it a GC-rich stretch. The GC-rich stretch allows for the production of GC base pairing in this region. And remember, the GC pairs are very strong because they have 3 hydrogen bonds between them. And then there's a little section that has, 6 to 8 A's. So there's a bunch of A's in a row. Okay. And both of these are important for the mechanism. And it's about the behavior of the enzyme under these conditions. So the enzyme has intrinsic properties that kind of check for the stability and the accuracy of the transcript being made. And so what happens is, here's your DNA down here, and the transcript is made, and here comes the enzyme moving along the DNA, and then it starts incorporating a lot of U's, and so that makes a bunch of AU pairs. And remember that the AU pairs are not very stable. And so the enzyme is able to detect this, and there's a built-in sort of automatic procedure where when this unstable base pairing is, if there's one AU or a couple of AUs, it's not a big deal, but when there's a lot of them in a row, the enzyme has a mechanism that goes, wait a minute, I wonder if this is all right. And so what it will do is it will back up. So that means it chews away these nucleotides, and it will go back. And the idea is it moves back to find a stable, a more stable base pair, like a GC, right? But because of the sequence here with this GC rich region, it has formed this hairpin structure, which is what this is showing here. And so what's happened is this has formed this hairpin, and so the enzyme backs up, but it gets stuck here. It can't go any further because you've got kind of like a brick wall here with this base pairing. And so it cannot go forward until the check has happened that says, is this the correct sequence? Where are the GC pairs that I'm kind of expecting? It can't go forward, it can't go backwards, and eventually there's just a mechanism that goes, okay, you've stalled here long enough. You have to fall off the DNA and go do your job elsewhere, right? So that's in a very, very simplified way what's happening. Now, when I talk about it, I always talk about it as if the enzyme kind of knows what it's doing. But remember, this is just an automatic feature of the enzyme. When certain unstable nucleotide pairs are detected, and there's a whole bunch of them in a row, there's a stopping and an attempt to back up. And when it can't back up, then it's unable to go through the check process and start, going back and continue with transcription, is the idea. And so it's kind of an interesting mechanism, and I think there's something that we will be talking about when we talk about regulation of operons quite a bit later in the semester. But this knowing about this will help you understand that mechanism too. It's another regulatory mechanism that depends on how the RNA that is produced folds up on itself. Okay. So that's the initiation, the elongation, and the termination part. And as I said, we're only covering the prokaryote part of it, but at this point, what's important to know is just that, remember I said at the beginning that eukaryotic RNA is transcribed in the nucleus and has to be processed properly and then transported into the cytoplasm. And we'll talk about the processing, the different things that happen. There's 3 things you need to know about that happen to that before it gets transported to the outside of the nucleus. And the genome organization is more complex, and we looked at this in lecture 2. So the genes are larger. They're spread out over larger regions. They have introns, those non-coding spacers. They have the 5' and 3' UTRs that are sometimes longer. They have more complicated regulatory regions. Sometimes they have regulatory sequence that is 1,000 or more nucleotides away from the gene. And the DNA is packaged in chromatin. So all the stuff that I talked about, lecture 2, the histones and all that, that is eukaryote packaging. And so think about the DNA wrapped around proteins. It's harder for the RNA polymerases to find and bind to the correct sequences if they're all wrapped around proteins. And so there's levels of regulation that that deal with how densely packed the chromatin is and so on. So there's much more stuff to think about in that sort of regulation. So these are the kinds of processing, though, to know about. So on the 5' end of that RNA, so the very first nucleotide that's added is a special G, and it has a methyl group added to it. So it has it's a chemically altered G, and it's required for stability. So there are nucleases, proteins in cells that digest or degrade RNAs in particular. That's why they don't last a really long time in the cell. And so having that extra methyl group on the guanine on the 5' end helps to prevent that degradation. And it's also required, it's part of the process of initiating translation. It's part of the recognition process that where the ribosome binds, where the ribosome binds to the messenger RNA. And then on the 3' end, after you've finished transcription elongation, there is a DNA sequence there that basically recruits an enzyme, and that enzyme comes and binds to the RNA, and it cuts the end off, and then it adds adenines, a whole bunch of them, 150 to 200. So it can be somewhat different for each copy of the mRNA for the gene. And again, this keeps it stable. If there's nuclease enzymes that are out in the cytoplasm of the cell, if you have a really long poly A tail, then you can be translated many times before the nucleases start chewing up the RNA, because these start at one end of the molecule, usually the 3' end, and they just remove nucleotide after nucleotide. So it's required for stability. RNA is inherently, naturally less stable than DNA is, and so these are little tricks that the cell plays to keep it stable long enough for it to do its job. And then there is splicing, and it's a complex process, and it's carried out by RNA protein complexes called spliceosomes. There's a lot of evidence that the actual catalytic part of a spliceosome, the part that is doing the cutting and pasting of the sequence, is actually the RNA part. RNAs have been known for quite a long time to perform catalytic reactions, just like protein enzymes do. And the splicing part starts as transcription is proceeding, so you don't have to finish transcription before you do the splicing. So there are special sequences that are the same at the edges of the intron, and the intron is the part that is removed, and the exon is the part that contains the messenger RNA. So if you remember GU and AG, that's enough. There's a bunch of other sequences that are highly conserved that we see in a lot of introns. But this will be good enough. And the idea is just that the spliceosome, which has, these are the little RNAs, and then these are the proteins that sort of hold the whole thing together. And notice that some of the RNAs are recognizing the GU on this side, and some of them are recognizing the AG on this side. And they make cuts, and then this part of the intron is attached to this A up here to make this weird lariat structure, which then gets degraded, and then these two exons are stuck together, right? So yeah, I think that's probably sufficient to know just that the DNA, the RNA, sorry, is cut to either side of these. The intron is it forms this unusual structure that then gets degraded, and exon 1 and exon 2 in this case are stuck together. And just another thing to note, that in eukaryotes, there's no introns in prokaryotes, but in eukaryotes, it's been known for some time that there are quite a fewer genes than you might expect for a much more complex organism. You know, think of humans with only about 20,000 genes or so. But it turns out that one gene can produce messenger RNAs that can be spliced in different combinations so that instead of having similar proteins in the brain and the liver and the heart and kidney or something, and have separate genes for that, you can have one gene and then you can combine the parts in different combinations to make a protein that is specific to brain, and another one that is a variant of that but is found only in the digestive system or something like that. It's kind of interesting. And this is just an example. So this is the primary messenger RNA transcript, and you can see, so the number here means the exon, so that's exon 1. And then there's two different options for exon 2. You could get 2A or 2B. Oh, here's an 1B, so you can have 1A or 1B. And then 3 and 4 and 5 are the same for all versions of this. And then you've got 6A and 6B, so you can have one or the other of those. 7 and 8 are the same all the time, but then you've got 4 different exons for 9. And so if you look at sort of striated muscle versus smooth muscle, you can see like they both have these ones. They both have 1A, but one of them has 2A and one of them has 2B, and there will be slight amino acid differences in the protein that gets made here. And then they both have 6B, 7 and 8, and then one of them has 9A and 9B, and the other doesn't have 9A or 9B, but has 9D, right? And so if you just have a quick look, you can see sort of the different combinations that are possible in different cell types. They will have different combinations of exons, and the proteins will be, they're all doing a similar job, but because the tissue is different and the proteins it interacts might be different, you build a just slightly different protein. There is a gene called DSCAM, which is Down syndrome related cell adhesion molecule, and there's a gene like it in flies that was discovered similar to the human version that's associated with Down syndrome, and it has something like 22,000 different possible. If you look at all the different exons and variants and stuff, and you think, okay, if we look at every possible combination of these, there's possibly 22,000 different proteins that could be made from one gene. And they certainly haven't identified all of them, but they've identified quite a large number. So it's kind of an interesting thing. And if you take MBB courses, you'll learn a lot more about this for sure. Okay, so that's the end of the second prep for this lecture. At the end of this process, we now have a transcript, and if it's a eukaryote, it's been processed and sent out of the nucleus. It's out in the cytoplasm. So what do we do now? How does this RNA molecule direct the production of a protein? That is the subject of the next set of lectures, and I'm going to probably divide it into 3 lectures. And so that will take care of the following week. And I hope you will find it of interest.